स्टार्ट करें क्या? Should I start the webinar? Welcome all the respected participants in the international webinar on the toll-like receptor 4 mediated immune response in the development of skin cancer. We are honored to have with us our chief guest, Professor Akhilesh Pandey, who is our Vice Chancellor and eminent bioscientist, and we are grateful to mention that he accepted our invitation in a very short notice and happily agreed to give a brief talk on mushroom as immunity booster. As we know, mushrooms are powerful pack of antioxidants and vitamins and known as disease-fighting stars. His speech will be very informative as we all are looking towards building our immune system in present stressful conditions. Our next speaker of this international webinar is Dr. Nabiha Yusuf, a well-known immunologist presently working on dermatology in Alabama University of Birmingham. She will going to give information on the role of toll-like receptor 4 in skin cancer. As we all know, toll-like receptors are molecular domains belonging to family of pattern recognition receptors that initiate signaling pathway resulting in cytokine storm and thereby initiating innate and cell-mediated immunity. Dr. Nabiha Yusuf has done extensive research on targeting toll-like receptor 4, which will be very informative towards development of therapeutic strategies to prevent skin cancers. Welcome, Dr. Nabiha Madam. I request participants who are unable to join Google Meet can join YouTube through the link we have sent for it. This is information for the participants. Before starting our webinar, I would like to request our principal, Dr. Bhardwaj, to speak few words and give us permission to start this event under the flagship of Pursuit of Research Excellence. Welcome, Dr. Bhardwaj, sir. Welcome, sir.
Thank you, Dr. Rekha Khanna. It's really a matter of great pleasure that today we are organizing an international webinar based on cancer research and causes of cancer. So it's really a matter of pride for this college that Dr. Nabiha Yusuf from Birmingham, she has joined us and has very kind sense to deliver a very important talk about the development of cancer and role of this toll-like receptors. So I welcome Dr. Nabiha Yusuf from Birmingham, and at the same time we are thankful to Dr. Akhilesh Kumar Pandey, who is मैं कहूँगा बहुत यशस्वी कुलपति हैं, हम लोग बहुत भाग्यशाली हैं. अखिलेश पांडे साहब जैसा कुलपति विक्रम विश्वविद्यालय को मिला. पहली बार मेरा भी शैक्षणिक अनुभव 30 वर्ष का हो गया, तो महाविद्यालयों को विश्वविद्यालय का अभिन्न अंग मानते हैं और जो ये विचार करते हैं कि महाविद्यालय के विकास उतना ही महत्वपूर्ण है जितना कि विश्वविद्यालय का विकास, और महाविद्यालय के विकास के साथ ही विश्वविद्यालय का विकास. हम लोग बहुत गौरवान्वित हैं ऐसे कुलपति जी को पाकर. इसके अतिरिक्त तो जैसा कि बात हुई, वे एक माइक्रोबायोलॉजिस्ट हैं, वे स्वयं भी विज्ञानवेत्ता हैं और आज इम्यूनिटी बूस्टर्स के बारे में हमसे बात करने वाले हैं और किस प्रकार से मशरूम जो है वो इम्यूनिटी बूस्टिंग का काम करते हैं, इसके बारे में वो बात करने वाले हैं. तो आई वेलकम both the guest speakers today. I must speaker is Dr. from Birmingham and VC sir is our chief guest today. So I welcome both of them and I congratulate Department of Zoology, Dr. Shaila sir, Dr. Pandey, Dr. Siddiqui, Mr. Simrat Vedya ma'am and Dr. Shubha Shoche to organize, and of course Ashib and my whole Department of Microbiology. They are all organizing a very good event today. I congratulate you all and I welcome all the participants in this webinar. Thank you very much.
Thank you.
Good afternoon. It is a privilege for me to give you a brief introduction about Dr. Akhilesh Pandey, our Honorable Vice Chancellor. Honorable Sir has worked as former chairperson of Madhya Pradesh Private University Regulatory Commission. Dr. Pandey is well known bioscientist and mainly worked on proteomics and genomics. He has published more than 650 research papers in national and international journals. Now I humbly invite Sir to deliver his lecture on mushroom as immunity booster. Thank you.
अरे सर
Sir, we are waiting for you.
Sir, we are waiting for you. जी, okay. Okay. जी. Connect. Dr. Pandey connect.
सर कुछ ही समय में हमारे साथ कनेक्ट हो रहे हैं
Dear all
Hello.
participants, yeah, shortly our Honorable Vice Chancellor is coming and joining to you within a period. I think that our Honorable Vice Chancellor has joined us. So please welcome, sir.
Hello.
Sir, you are audible.
हमारी आवाज आ रही है आपके पास?
Yeah, sir, you are audible to you.
नहीं आवाज आ रही है आवाज.
You are audible to us.
हेलो.
सेंटर में नहीं पहुंच गए सेंटर में
हेलो. सर की आवाज आ रही है?
सर आपकी आवाज आ रही है हमको.
अच्छा मैडम, ठीक है मैडम, बताइए हम शुरू करें.
जी हाँ बिल्कुल सर, स्टार्ट करिए सर. जी वेलकम सर.
Today in this webinar, first of all, I would like to thank Dr. Bharadwaj Sahab, Madam Soila, and almost all the participants. I welcome on behalf of the Vikram Vishwavidyalaya. Today is one of the most important and burning topic of society, that is a cancer. Cancer is one of the most disastrous disease in India, especially in the world, but especially in India. Cancer, day by day, this disease is increasing day by day. There are some problems which is directly related with the increase of the cancer. One of them is a use of chemicals. As you know, earlier there was a balance between the nature and the Sanskriti culture, but because of the ignorance of our culture, especially Indian philosophy and culture, we forgotten our most of the traditional way of lifestyle, working, and most of them, one of the most important thing is agriculture. Earlier in India there was a animal based agriculture, but due to huge requirement of food grains, our earlier administrators, they have suggested a committee that is a green revolution, and because of the green revolution, there is no doubt about the we increase our production enormously. But within the next 60 years, when we incorporated the number of chemicals, they accumulated in the field, and ultimately through the food chain, the chemicals reach to our body. And nowadays synthetic chemical is one of the most important challenge for the human health. That is they are also responsible for the cancer. 1960s, that was the golden period for the chemical herbicides, pesticides and chemicals. But if you just remember one of the places in Punjab is considered one of the most important, most developed, advanced agriculture. But one of the train, that is a cancer train, started from the Kapurthala and reached to just run in between the Kapurthala and this Surat. Most about 90% of the patient, they are suffering from the cancer. Why? Because of the use of synthetic chemicals. These synthetic chemicals, when reach to our body, especially due to this magnification, this bioaccumulation is one phenomenon, and then when they reach to our body, they become more just bio magnified. When they become bio magnified, they release certain for example, if you just take one example, DDT, which is banned long back, banned in the Italy, where the scientists have they got the Nobel Prize in the development of this pesticide. But in India, still we are using DDT. And DDT, when reached to our body, it convert into DDE. DDE is highly responsible for mutation in our cell, especially it is a mutagenic. It is considered as one of the most important mutagenic chemicals. Similarly, there are so many other chemicals used as a pesticide. They are mutagenic, highly mutagenic, and still we are using these chemicals, and we forgotten our organic farming, which was very safe for human health. Another things which is very important, that is environmental problems, which are contributing in the increase in the cancer cells, especially some of the this inorganic based chemicals released in the atmosphere. They ultimately also reach to our body, and they become a mutagenic. There are some other chemicals like tobacco. If you just take the examples of the report of the oral surgeons, you will find the tobacco, especially in the rural areas, tobacco, this BD are just like some of the drugs. Not only tobacco and BD or others, but even the alcoholic products, they are also causing a mutation in our cells, and ultimately they are converting into this cancer cell. In women in India, there are two type of cancers, very common. One is breast cancer, and other is uterus fibrosis. Uterus fibrosis and this breast cancer is also because of the lifestyle. There are some food habits. Food habits are obesity. Obesity ultimately related with the food habits and health fitness. That is also one of the contributor of the this cancer. Even lifestyle nowadays, if you just remember yoga or meditation, stress is also contributing in the development of cancer. Stress, sometimes stress directly responsible for release of there are some chemicals which ultimately cause the this damage to our cells, and responsible for mutation. So these are some of the problems which is contributing heavily in the increase of the cancer cells, cancer, ultimately cancer diseases. But prevention is better than cure. We should think about the prevention of the cancers. And one of the most important thing is food habit. Food habit, and there are some chemicals which are immunomodulators. Immunomodulators are anti-cancerous products which can prevent these cancer cells. One of them is this. I am actually a botanist. I'm not a zoologist, or I'm not work on the cancer, but there are some products, natural products like mushroom. Mushroom, if you consider, mushroom is known as a complete food component, and there are many chemicals because sometimes cancer also created by this malnutrition. Malnutrition is also contributing in the enhancement of this cancer. So mushroom, which is very natural product, which is available in the nature, it can be used for the prevention of this cancer. One of the example is, if you just consider vitamin D. Vitamin D is also very important for the enhancement of immunity. Vitamin D for non-vegetarian is available in the meat, but for vegetarian peoples, mushroom is the only source of vitamin D, or you can say in sunlight. But most of the time, people ignore the consumption of mushroom. If you consume the mushroom, it can enhance our immunity, and immunity ultimately related to the cancer cell. So organization of this conference, this webinar is very important, especially to understand the mechanism of cancer, how cancer cells produce, how it multiply, or how it can prevent. Until unless we know about the exact mechanism of the cancer, this mutation, we cannot go for the prevention, or we cannot go for the management. So I congratulate the organizers, especially Principal Sahab, Madam, for organizing this webinar. And see, this in webinar, one of our alumni in Rani Durgavati Vishwavidyalaya, she is also participating. I think she is joined the webinar. She is the product of our department in Rani Durgavati Vishwavidyalaya, Jabalpur. I thank her for sparing her time, valuable time, to just create awareness in our peoples and our students. Thank you very much.
हाँ मेरे से
Hello.
Thanks, sir. Myself, Dr. Shaila Issa. I extend my gratitude and thanks to Vice Chancellor, Vikram University, Dr. Akhilesh Pandey, sir, for giving us their time for this webinar. Sir, you give source of immunity booster in coping up for this pandemic
ओके सही बात है बहुत साउंड
of
हो रहा है
corona. Thank you very much, sir. Our next is Dr. She belongs to us, know already. Doing PhD, completed her PhD from Aligarh Muslim University. Since 2020, he served on different posts in Birmingham University. When I see the biodata, it's too long to explain in in International Journal. He is not only dermatologist, epidemiologist also, and mentor of so many places. Faculty of Neuroscience Program, Faculty of Scholar Program, Beckman Scholar Program. She also work as in the Cancer Prevention Center. She is a very good, I say it's a bel esprit, means very intelligent, very sincere, hard working since young age. Nowadays, she is doing a good time with us, and I am very happy that she give their time for this particular webinar. It's an international webinar, and I am also thankful, Dr. Pandey, because both of the student of bioscience department, both of the one is the faculty alumni and one is the student alumni of bioscience department of Jabalpur. She also gained so many awards and honors from USA, so many theses submitted under the supervision of Dr. Nabiha. His major research interest is on dissect the role of innate immune response, induced cutaneous DNA damage. In the Department of Defense, she also continued to perform translational studies on TLR4 inhibitor in prevention of skin cancer. So he received so many funds from different departments of USA. And it's very good for us. So again, I invite Dr. Nabiha Yusuf, please start their talk and acknowledge us. Thank you,
Dr. Sheila, and everybody in Ujjain. Thank you, Dr. Pandey. It's good to see you after such a long time. I remember you as a student when I was a student at Rani Durgavati University, and I'm like really grateful to all of you for inviting me to give this talk. And I hope this is not really relevant too much to India since it's skin cancer, but I think the same principles can be applied to different cancers because these signaling pathways are common across the board. So let me share my screen. So can everybody see my screen?
Yes, ma'am.
Yes, ma'am.
Okay, great. So, as you all know, I've come from US, so I might just, give you some data regarding US incidence and, about skin cancer. So we don't really see much of skin cancer in India, though, but in US, it's a major problem. And if you see the global picture here, this is a map from a global map from 2000. This is from 2020, right? Like just last year. And this is 2060. So you can see how the UV index is increasing. And if you look at the picture from like around after 40 years from now, it's going to get really bad. And this is a map of US, and this is where our university is in Alabama. You can see it, it's in the red. So we have very high incidence of skin cancer. That's why it's very important for us to look at mechanisms. Now, you all must have heard about UV radiation and the UV spectrum and how many types of UV are there. So I'll just give you a brief idea about the types of UV and what they do. So mainly there are like the three types, UVA, UVB, and UVC. They fall under different spectra. And UVA, as you can see here, it can cause premature aging, wrinkling of the skin, and implicated in skin cancer. But UVB mainly causes skin cancer, sunburns, and cataracts. And UVC is extremely dangerous, and a lot of it is filtered, is stopped by the ozone layer we have in the atmosphere. And now we talk a lot about pollution and global warming and all those things. So that is depleting the ozone layer. So we are at a really high risk of getting UVC too. So the type of skin cancers, so there are like different types of skin cancer. Mainly we have like the two main classification is like the non-melanoma and melanoma skin cancers. And the non-melanoma skin cancers can be divided into squamous cell carcinoma and the basal cell. And then melanoma is separate. So you can see here, this is the squamous cell carcinoma here. And this is the basal cell. It arises from the basal cells here, and this is the melanoma. So this is like the clinical picture, how it looks like in the patients. This is the basal cell, picture of basal cell carcinoma, and this is squamous cell carcinoma. And this is the typical picture of the skin, which is divided into epidermis and dermis. So this is the uppermost layer of the skin, and this is the dermis. And you have all the hair follicles. These are all the hair follicles. And this is all the sebaceous glands here, sweat glands. So non-melanoma skin cancers can be divided into basal cell carcinoma and squamous cell carcinoma. So basal cell carcinoma is more important. It comprises of 80% of all skin cancers, but it is rarely fatal. And 50% of patients will have another lesion within 5 years. And it usually occurs on sun-exposed skin. And it's linked to intense UV exposure in childhood and adolescence. So we do not see a lot of incidence of skin cancer in India, but in US, people have light skin and they are exposed. Their skin is very sensitive. So, and they go a lot to the beaches, and also skin cancer doesn't happen all of a sudden. It's like the cumulative exposure over the lifetime. So you are exposed several times to UV rays. And when you grow older and your immune system breaks down, you are at risk of developing skin cancer. Now, the squamous cell carcinoma comprises of like 16% of all skin cancers, but this can be metastatic. It can occur on sun-exposed sites, and it's also linked to lifetime exposure. Now, this is a model for induction of skin cancer by UV. So like Dr. Pandey mentioned, cancers can be caused by several agents. So even skin cancer can also be caused by several agents, which includes chemicals. So we do a lot different kind of research in my lab, and one of that is also looking at chemical carcinogens and development of skin cancer, especially polyaromatic hydrocarbons from, like the oil, burnt oil and all that, barbecue. But in this talk, I'm just focusing on UV-induced skin cancer. So this is to give you an overview of how skin cancer happens. So this is the sun, and you can see when it falls on the skin, it causes damage. It causes DNA damage, and we will see later how it does that. And that DNA damage, it causes mutation in the tumor suppressor genes like P53, PASH, or it can cause activation of oncogenes like KRAS, HRAS, and NRAS. Now, UV can also cause immune system deregulation. It can cause certain immunosuppressive cytokine release. It can cause Langerhans cell depletion. Langerhans cells are the dendritic cells that are present in the skin. And so what happens when all this damage occurs, usually the cell undergoes arrest and it's repaired, and then the normal cell proliferation takes place. But if these things do not take place, if these mutations are not altered, the DNA damage is not repaired, it can cause immune suppression and abnormal cell proliferation. And as a result, we can get tumors. So UV is primarily immunosuppressive, and individuals who have suppressed cell immunity have an increased risk of developing skin cancers. So what does this mean? That means like people who are who undergo organ transplants, or they are on immunosuppressives for some other reason, they have a high risk of developing skin cancers. And UV-induced damage in the form of cyclobutene pyrimidine dimers is one of the earliest molecular events in UV-induced immunosuppression and skin cancer. So you can see here what UV does. So UVA can cause mainly oxidative stress, and it can cause DNA damage in the form of 8-oxoguanidine. And UVB primarily can cause CPD, cyclobutene pyrimidine dimers, and 6,4 photoproducts. So we're going to focus on CPDs in this talk. So you can see the adjacent thymidines here, they get linked and they form like a ring. That's why it's known as like cyclobutane ring, and this dimer is formed. So what it does, it stops the normal proliferation of cell, and there's a mechanism in our body to repair it. But if this does not get repaired, it can cause abnormal cell proliferation. So one of the mechanisms to repair this is the nucleotide excision repair, and it removes DNA damage by two pathways, transcription couple repair or global genome repair of DNA. So you can see here the pyrimidine dimer is formed here. And then this is the incising activity. It cleaves that part of the DNA, and then you can see it excised. This part is excised, the damaged part, and then the polymerase introduces new bases, and the ligase, of course, seals it up. So you can see this whole thing got repaired. Now, moving on, what we study. So we study toll-like receptors. Probably people in microbiology must be very familiar with toll-like receptors. So toll-like receptors were initially discovered in Drosophila, and they were studied in context of infection. And toll-like receptors, you can consider, I always tell in my talks, like you can consider toll-like receptors like a toll on the highway. So when you have to go pass through the toll, you have to pay some kind of tax, and it will not allow you to go through like freely. So similarly, these toll-like receptors, they're known as pattern recognition receptors. So they're very important component of innate immunity. So what is innate immunity? Innate immunity is very non-specific. It's not like adaptive immunity, and it recognizes patterns. It does not recognize any specific like antigen or anything, just patterns. So this is like one of the members of the innate immune receptors. So they recognize patterns. So suppose there's a microbial attack or something, so it will be recognized by the toll-like receptors. The signaling pathway gets activated, and that is taken care of. So there are several toll-like receptors in our body. And so there's some minor variation between mouse and humans, but mainly there are like TLR 1 to 13, and we study mainly TLR4. So why we study TLR4? TLR4 is unique because it is recognized. So you know how the receptors are activated. The receptor is activated when a ligand binds to the receptor, then the signaling pathway is activated. So the unique part about TLR4 is it is recognized by several exogenous and endogenous ligands. So one of the common ligands for TLR4 is actually LPS, or lipopolysaccharide, which is present in many gram-negative bacteria, and that actually activates TLR4. But so all this study on TLR4 started with infection, but we started studying something different. We looked at so the infectious agents are known as pathogen-associated molecular patterns. So there were some molecular patterns in the pathogens that were recognized by the toll-like receptors. But then the research moved on, and then people started looking at damage associated molecular patterns. So these were non-sterile. They were not infectious agents. They were like something like when you give chemotherapy to the patient, so the cancer cells release certain molecules like HMGB1, or like sometimes heat shock proteins are activated. So they can all activate TLR4. So this is like a new area that started after the people thought they were only restricted to microbes. And now it's a very like developed field. So this is our molecule of interest, TLR4. We study this, and you can see there's a picture which gives you an idea about all the most of the toll-like receptors and their ligands. And then we thought, okay, what happens, which is the pathway? There are two pathways. So TLR4 signaling signals through the MYD88 or the TRIF pathway to activate NF-kappa B and produce cytokines. So we wanted to see which pathway is taken by TLR4 for this process. So these are two pathways here. Either it takes the MYD88 pathway or the TRIF pathway. So we did some staining here. We took the wild type mice, which is normal mice, and we took the MYD88 knockout mice and the TRIF knockout mice. So all these mice are on the black background, black 6 background. We exposed them to UV, and then after 24 hours, we looked at the cyclobutane pyrimidine dimers. You can see these green dots here. And this is the wild type. So even in wild type, there's a mechanism. So they do get repaired to some extent. And in the MYD88 knockout, you can see when you knock off MYD88, they're completely repaired. So this is some MYD88 is doing something to stop this repair process. But when you knock off TRIF, you get a lot of CPDs, which means that this process mediated pathway and not the TRIF pathway. So we also decided to look at the XPA gene, which is the DNA repair gene. And you can see here it's highly expressed in the mice when you knock off MYD88. And when you knock off TRIF, it's decreased. So let me tell you what XPA is for people who are not familiar with the term. It's called the xeroderma pigmentosum complementation group A gene. So xeroderma pigmentosum is a disease. It's a rare genetic disorder where people have a deficient DNA repair capacity. So these people cannot go out in the sun. If they are like exposed even for a very short time, they can get skin cancers. And actually, in US we have read like news reports and seen people like that who have this kind of condition. So these mice kind of mimic that disease. So for this part, we can conclude that the UV-induced CPDs were repaired more efficiently in the skin of TLR4 knockout mice than in the skin of wild-type mice. There was significantly higher expression of the XPA gene in the skin of TLR4 knockout mice than the wild-type mice. And the UV-induced CPDs were repaired more efficiently in MYD88 mice than TRIF. And XPA was also upregulated in MYD88 knockout mice than TRIF knockout mice. So moving forward, we want to see what is like, how does UV cause immune suppression? Because I mentioned a little bit earlier that DNA damage leads to immune suppression. So you can see here, even like a small dose, you don't have to have like a really high dose or go on the beach to get like high dose of UV for immune suppression. Even low dose of UV can cause immune suppression. You can see from this drawing. And this is the reason why people on a cloudy day or people are very careless about this. They say, okay, this is it's not very hot. Maybe we should just ignore sun protection and all that. And that's the way, in that way you can get immunosuppressed. So how did people think that immunosuppression can cause cancer? So there was a lady, Margaret Kripke, she's still there in MD Anderson, and she did a very classical experiment to prove that immunosuppression can cause cancer. Earlier, people thought, okay, most of us know that, okay, inflammation can lead to cancer. Okay, that's true. But in this case, immunosuppression has a big role too in causing cancer. So she did a simple experiment. So you can see from this, she took 3 panels of mice. In the first panel, what she did, she exposed the mice to UV. So when you expose the mice to UV for a long period of time, they develop tumors from their skin. So she took that tumor and she transplanted into a normal mouse, with no knockout, nothing. And this mouse, when the tumor was injected, it got rejected. So why did it get rejected? Because this mouse had a normal immune system, which of course took care of the tumor, and the tumor was gone. But in the next panel, what she had, she gave some immunosuppressive drugs to the mice. So as a result, the immune system was suppressed. So when she injected the tumor in this mouse, the tumor grew progressively because of the suppression of the immune system. So then what she did in the last group, she exposed the mice to UV before injecting the tumor. And you can see here the tumor grew. So you can see from these two, like the UV treatment is kind of mimicking the treatment with immunosuppressive drugs. So that proved that UV is immunosuppressive. Now, what we do, we study a very simple model. If you are familiar with delayed type of hypersensitivity, which is, so we use this model. So in this model, basically what we do, we use a hapten. So hapten is a chemical, hapten, DNFB, dinitrofluorobenzene, and we apply to the skin of mouse. You can see here on the back of mouse. We shave, every time we do these experiments, we have to shave the hair. So, and then we'll look at this contact hypersensitivity or delayed type hypersensitivity response. So when you expose this, so normally if you just apply a hapten and you measure, so when you apply the hapten on the back, we measure the ear of mice. After like 5 days, you will see later how we do this experiment. So this is a normal contact hypersensitivity response. The ear swelling will take place. The T cells migrate to the lymph nodes, and then you challenge the ear another time and you get a big response. So the ear swells up. So this is a classic contact hypersensitivity experiment. But when you pre-treat the mice with UV and then you apply this chemical on the skin, you see there is no ear swelling. So this is a suppression of this response. Now, interestingly, when you even when you apply, like expose the mice to UV and apply DNFB and even apply DNFB at the remote site, you can see suppression of contact hypersensitivity. Now, this response is antigen specific. So when you apply, when you inject, so what we do, we expose the mice to UV and then apply DNFB. And we take out the spleen and then we isolate the T cells from these mice and inject into two groups. In one group, we again expose to the same antigen, which is DNFB, hapten, and then you don't get a CHS response, which means that this thing has memory, you know, like this response can induce memory, and you can get like, it still recognizes the antigen and there's no CHS response. But if you use an unrelated antigen like oxazolone, you can see a normal CHS response, which means that this response is antigen specific. Now, how does that happen? Like I mentioned before, UV radiation falls on the skin, and you can see this contact allergen is applied on the skin, on the cells. And these cells, they migrate to the lymph nodes, a migration of Langerhans cells from the skin to the draining lymph nodes. And in normal condition, without UV, they will activate the effector pathway. For example, they will present the antigen to the T cells, and there's increased expression of these molecules and normal response. But when there is damage, when they go to the suppressor pathway, they can induce suppression. So the reason I gave you this little background is because we're going to study, I'm going to show you data, our lab data on immune suppression. So how we did these experiments. So this is an experiment, how we do this. So this is like the mouse, we expose them to UV. And on day 0, we applied this chemical, DNFB, on the back. And after 5 days, we applied on the ear. And before we apply in the ear, we measure the baseline thickness of the ear. And then after application of antigen, we measure the ear thickness for 3 days. So the whole principle for this is like when the antigen first reaches the lymph nodes, it generates T cells, it generates memory cells. So when a second encounter happens, like in this case, when you apply it on the ear, the cells recognize this antigen, and as a result, they migrate to the site and then causes massive infiltration. And that's how the ear swells up. So it's a very simple, very classy experiment to study immune contact hypersensitivity. So Now we wanted to look at TLR4. So we again took the TLR4 knockout mice and the mice which had TLR4. And then we did this immune suppression experiment like I showed you in the model. So you can see here in the normal case what will happen. This is just the DNFB is a positive control with just the chemical. And in this group, we pre-expose the mice to UV and then we apply DNFB. So you can see there's a significant reduction of ear swelling. And but in the case of TLR4 knockout, you can see there's hardly any difference. It's not very significant. So TLR4 is somehow involved in this process. And we do a HNE staining of the ear. We cut the ears and we stain them. So you can see here, this is the naive, this is the DNFB. And you can see after treatment with UV how this has gone down. The swelling, and this is in the TLR4 knockout, which we can hardly see any difference in ear swelling. Now we wanted to do the other experiment where we wanted to look at the adoptive transfer, whether these cells can be transferred and they can still cause immune suppression. So here we apply DNFB on the back, and when we isolate purify the regulatory T cells, and we inject it into the mice. And then we again challenge them with the ear, and then we measure the ear here. So you can see here in the wild type mice, even when you transfer the cells, they can still cause immune suppression, but you do not see that in the TLR4 knockout mice. So we did some flow cytometry to look at these cells. So this is like the flow cytometry picture. This is the CD4 cells, CD8 cells. So we can see here the CD4 cells. These are the CD8 cells. They are like reduced in the wild type mice, but they are consistently present in the TLR4 knockout mice. Similarly, you can see the Tregs, which are increased after UV treatment in the wild type mice, but not that TLR4 knockout mice. Similarly, so these regulatory T cells that I mentioned, they are CD4 positive, CD25 positive, and FOXP3 positive. So FOXP3 is a transcription factor for these regulatory T cells. So we purified this T cell population that I showed you earlier. And then we gated them actually on CD25 and FOXP3, and as you can see here, FOXP3 is increased slightly, but it's increased. These are the double positive cells, CD25, FOXP3, so they're increased in the wild type mice, but they're not increased so much in the knockout mice. Then we also looked at cytokines. So these cells are immunosuppressive. So they produce some immunosuppressive cytokines like IL-10 and TGF-beta. So you can see here how these cells, this is IL-10 on this axis, and this is TGF-beta. So you can see here how this is significantly increased in the wild type mice compared to the knockout mice. Then we also looked at, there's a big interest nowadays in looking at myeloid CD11B GR1 positive cells. These are like a population of myeloid cells, and they're very important in immune suppression. Especially people with cancer have a lot of these cells. They're more commonly known as MDSC, or like myeloid-derived suppressor cells. So we try to look at those. And you can see here they are increased significantly in the wild type mice, but not in the knockout mice. Now we also wanted to see, like in the first part, that we wanted to see which pathway is used for this immunosuppression. So this is the wild type mice. So when we do that contact hypersensitivity experiment, we measure the contact hypersensitivity at different time points. It's like a 3-day measurement. So you can see here, this is the positive control, DNFB, and this is the UV treated. So you can see there's a significant reduction in ear swelling. But if you look at MYD88 knockout mice, there is no significant difference. So MYD88 has a big role to play here, as you can see this. because it's completely resistant to UV-induced immunosuppression when you knock off MYD88. And if you look at TRIF knockout, which is the other molecule in the pathway, you can see that it goes back to similar to what you see in the wild type mice. So we believe that TLR4 signaling through the MYD88 pathway to cause immune suppression. Now, we also did more flow cytometry to look at these same cells, which I showed in the TLR4 knockout in MYD88 knockout mice and in the TRIF knockout mice. So you can see here, these cells, CD4, CD25 cells were increased in were decreased in MYD88 knockout mice, but they were increased in TRIF knockout mice, similar to wild type mice, and they're pretty consistent here in the MYD88 knockout mice. And this, we also looked at the FOXP3 cells and IL-10 producing cells. So these also are pretty consistently low when you knock off MYD88 compared to the wild type and TRIF knockout. Similarly, the CD11B GR1 positive myeloid cells were also pretty same in the MYD88 knockout mice before and after UV compared to the wild type and the TRIF knockout mice. So from this part, we conclude that TLR4 deficiency prevents UV-induced immune suppression. And this can be transferred by regulatory T cells in mice. And the TLR4 deficient mice have a lower FOXP3 expression. They secrete less IL-10. And this is MYD88 dependent. And regulatory T cells from MYD88 deficient mice have lower FOXP3 expression and they secrete less IL-10. And now we wanted to look at role of TLR4 in UV-induced tumor development. So this is our tumor cutaneous carcinogenesis protocol. We actually expose the mice to 200 millijoules of UV, and then this is an experiment of like 40 weeks, almost takes a year, and then we examine for tumors on the skin. So we compared the TLR4 deficient and mice which had TLR4 here. So you can see when you knock off TLR4, you kind of reduce the number of tumors in mice, and this is the wild type mice. And here there were a lot of mice, percent tumor-free mice here. And you can see even the tumor latency was lower in the knockout. So tumor latency is like the time the first tumor, you can start seeing the first tumor in mice. And these are like skin tumors, so they're very easy to count because you can see them visibly, like you see them with a naked eye. So what we do, we kind of document them. We create sheets where we record each and every tumor. So then we took out spleens from these mice after we sacrificed them after 40 weeks. And then we again looked at CD4, CD25 positive cells. So this panel is like TLR4. This is a wild type mice. So you can see here the regulatory cells were increased compared to the TLR4 knockout mice, where you hardly see an increase. And we also looked at FOXP3 expression, so which is really diminished in the TLR4 knockout mice. Similarly, we looked at the other suppressor cells, CD11B positive, GR1 positive cells. So these are also like increased in the wild type mice, but there's hardly any increase in the knockout mice. So from this, we concluded that the TLR4 proficient mice, or mice which had TLR4, were more susceptible to UV-induced carcinogenesis than the deficient mice. And they were significantly fewer tumors in these mice, and they remained tumor-free for a very long time. The CD4, CD25 regulatory T cells from these mice produce greater amounts of IL-10 and TGF-beta than the cells from TLR4 deficient mice. And expression level of FOXP3 in the skin was greater in TLR4 proficient mice than in the deficient mice. And the CD11B GR1 positive cells from UV-induced proficient mice produce significantly greater amounts of nitrate and arginase. So I have not shown this data, but these are some of the markers of the myeloid-derived cells. Then CD11BGR1 cells from TLR4 deficient mice. So this study has been going on for some time, and then we thought, okay, we need to look at an antagonist which we can take to the clinic. And dermatology, in the field of dermatology or like skin, topical inhibitors are very popular because you can apply them as a sunscreen, mix them in a cream and apply them. So, and they can be really easily done in the clinic. So an antagonist was developed somewhere in Japan. It's called TAC242 or Receptor, and we decided to look at its role in regulation of UV damage and tumor development. So this is an ongoing study we have seen. So when so in this case, we take normal mice. There's no knockout mouse here. We take normal mice and we apply this inhibitor with and without UV. So these are the controls, and this is UV 24 hours, 48 hours, and you can see here. We wanted to see if this inhibitor, which like this suppressing TLR4, whether it can increase the DNA repair gene expression, XPA expression in the skin. So this is what we found. Then we wanted to confirm this. Okay, it's increasing XPA, so we want to like double check it. So we also have XPA knockout mice. So these mice are deficient in this XPA. So this, when we apply this to the wild type mice, so you can see this is repaired very efficiently. So these are the CPD. So these are the cyclobutene pyrimidine dimers in the skin. So when we apply this to the skin, this TAC242, you can see there's a reduction in these CPDs. But when we apply it in the XPA knockout mice, because these mice do not have a repair mechanism, right? So you can see there's no difference. That means that this TAC242 needs the DNA repair, the XPA to function and to repair the damaged DNA. So we also did an experiment, a tumor experiment with this inhibitor. So we used this TAC242 on the mice, and you can see here, after application of TAC242, there was a significant reduction in the number of tumors. So we also looked at CD4, CD25 cells like we did before, and we see there's a reduction after we applied the TAC242. This is in the lymph node, the first panel, and the second panel is in the spleen. And this is similarly with IL-10, which is immunosuppressive cytokine here. And this is the CD11BGR1 cells here. So consistently what we are getting with the knockout mice, we were able to show that with the inhibitor. And then we also looked at the myeloid cells in the tumor, the CD11BGR1 positive myeloid cells. So we see when we compare it with the vehicle, you can see there's a reduction in the CD11BGR1 positive myeloid cells. So we concluded from this that TAC242 treatment reduced the number of UV-induced CPDs via NER mechanism. There were significantly fewer tumors in TAC242 treated mice, and there were fewer suppressor cells like the CD4, CD25, and they produced less IL-10 and TGF-beta. And there were significantly fewer CD11BGR1 myeloid cells after treatment. So this gives you a picture. This is a proposed model for prevention of induced UV-mediated responses by TAC242. So this is like the whole, I'm trying to wrap up the whole lecture in this. So this is how UV falls on the skin, and the damaged cells, the dendritic cells, they migrate to the lymph node. This is the lymph node. This big thing is the lymph node. And here you can see the T cells here. So when they go to this, there's like a, they are damaged, so they are not able to mount an effector response. So it will increase the number of regulatory T cells here. It will decrease the effector or the CD8 T cells, and immune suppression will increase, and as a result, we'll get more tumors. On the other hand, in the absence of TLR4, or when you apply a TLR4 antagonist, what will happen? These cells will not be damaged, and they will effectively present the antigen to the lymph node, and you can get less regulatory cells and more effector T cells, and as a result, you get fewer tumors. So this work was done over a long period of time, and there were several people who contributed it. Some of them have graduated from my lab, like Asif is still around. He's the only one around, but we have Star Tehseen here. Star is at UAB right now. He's doing another postdoc. Tehseen is at Emory. And Heba, she was a veterinarian. She did some flow with us. Wesley is joined a company, and Purushottam is still around. And Ava, they've all gone into residencies. They've become physicians now. So a lot of this work was done. And this is my mentor. I did my postdoc with him, and we are collaborators now, Dr. Helmets. And I was funded. I got a career development award from Dermatology Foundation to start this research. I got some seed money from UAB Faculty Development Program and the Skin Disease Research Center. And I was funded by the National Institute of Arthritis and Musculoskeletal and Skin Diseases, which is still going on. And then the Dermatology Foundation, and Eva got a grant, a medical student grant from American Skin Association under my mentorship. So all these people, I would like to thank them for their contribution to this story. And this is where we are. And a lot of this is unpublished. So I would request you to not make it like public. So we are in the process of publishing those papers very soon, probably first half of this year. So thank you for your attention, and I'm open to answering any questions that you may have.
Dear all participants, the topic is open for discussion. If anybody is having a query, they can ask their query.
I have a question, ma'am. Have you seen any role of vitamin D in mediated?
Can you repeat, please? I think it got cut off.
Yeah, my question is that is there any, because this UV radiation is also have a role in vitamin D synthesis.
Yes.
So does that vitamin D has any role to play in the effect of this your MyD88 molecule mediated signaling?
Yes, it does. It does. So it depends. If you're talking about skin induced, like vitamin D can be produced by the skin, and you can also take it like, exogenous by orally or whatever, by supplements. So vitamin D, the role is pretty controversial. There are some reports where they say like vitamin D actually suppresses TLR4 signaling. And yeah, but it's not really concrete. And this is one area I'm really interested to work on to see. And I have another talk, I'm not showing it here, where we look at like vitamin D polymorphisms in skin cancer population, but we haven't really studied that role of vitamin D as such.
Thank you.
Yeah, no problem.
Any other participants who needs to fulfill or form their queries? Okay. So thank you all the participants, and thanks Professor Nabia and our Honorable Vice Chancellor for having such a nice talk on this session. In the last, I would like to say that other than any other professional and respective post, a scientist is a scientist who can fill the gap between the hidden and unhidden aspects of the nature, which is related to the research. Nature has long been an important source of inspiration for the medicine. and which possess enormous therapeutic potential, amongst other things regarding the treatment of cancer. As this was our opportunity that Professor Nabiha has highlighted the aspect on the toll-like receptors which is involved in the development of the cancer, skin cancer especially, and our Honorable Vice Chancellor has also highlighted on the aspect related with the medicinal mushroom. He has also highlighted that importance of vitamin D in the treatment of the cancer. So, in this concluding remark, I would like to say that as our invited speaker has highlighted that involvement of the ultraviolet rays, how this is involved in the development of the cancer.
I think the voice is getting cut. You need to probably everybody needs to switch off their videos. I guess that's causing a lot of um, it's slowing down the Google Meet.
Thank you, Nabia, for giving. Thank you, Nabia, for giving a nice, nice and comprehensive, detailed knowledge of the cancer.
You are welcome.
Cancer.
There was a question in the chat box about MTT. Can I talk to Riddhi Maheshwari? She here?
Why no voice is coming?
Yeah, I think.
Thank you very much, madam. It was very interesting talk, and really, I found some new interesting points to look after. And
Yeah.
I'm looking forward to read your paper, which in the forthcoming
Yeah,
paper.
two of them, actually, three of them have been published. We are publishing the TAG 242 sometime before June, hopefully. It's being written up. So there's more. There's a lot more to the story, lot of data, but I was not sure how much time I will have to present that. So I just presented like some important highlights of our research.
Certainly, when we present, this is the problem, that which one to present when you have a lot of data and
Yeah,
so many knockout mice and look.
so this is one of our projects. So we have several projects. The other project is looking at type 1 interferons, which is going on currently. So we are looking at type 1 interferons and their regulation in UV-induced cancers. Not only UV-induced, we are looking in cervical cancers, any of the infection-associated cancers, and autoimmune diseases also. Because my PhD was in autoimmunity, it's on cardiac autoimmunity. So I'm also interested in autoimmunity. And then we have some inflammatory skin diseases which we study, like hidradenitis, psoriasis, and I do a lot some epidemiological study also, like looking at biomarkers in populations and how they, like correlate. I'm interested mostly in the GWAS, EWAS kind of studies. Pharmacogenomics is my interest, current interest. I'm looking in those areas. So we have multiple projects. So this is one part of what we do basically. So, and it was nice meeting you all, and I hope we get to meet in person sometime. And it was wonderful. And feel free to type your questions in the chat box if you have, or you can just, I can stick around for some more time if you want.
Thank you, Madam Nabiha.
Welcome.
Thank you all of you for giving such a nice lecture, very informative lecture. Really, it is very interesting.
Thank you.
Thank you, Madam. Thank you all of you.
Thank you.
Thank you.
आपका माइक ऑन हो गया और इससे आपका वीडियो अब जब आपको बंद करना है तो नीचे उन्हीं का पे ही माइक और वीडियो बंद कर लीजिए।
Hello. Hello.
Hello, ma'am.
I am Dr. Suchitra Banerjee from IEHE Bhopal. I really would like to thank Dr. Nabiha Yusuf to present this very paper. It's very very important, and the information she gave is very, very informative, and it is very useful. And I am very happy to meet my colleagues like Rekha. I could see you. Everybody, I wish everybody a very happy new year. And I once more thank for this organizing such a nice webinar. Thank you.